Okay, in this video what I'd like to talk about now is the regulation of cholesterol synthesis. Now, if you recall the pathway for cholesterol synthesis, we are going to be taking several acetyl-CoAs, combining them into our HMG coenzyme A structure. And then the next step will convert into mevalonate by the HMG-CoA reductase enzyme, this being a redox reaction. And what we're doing in this process is using an enormous amount of energy. The amount of energy that is used is, of course, going to be derived from the fed state, but constructing the cholesterol ring, the four-ring structure with the tail and the hydroxyl group on it, that consumes an enormous amount of not only energy to put into this, but also reagents. Now, the problem with cholesterol is a little different than the problems with fatty acids. When we construct fatty acids from our acetyl-CoA, the fatty acids go into storage. And then from storage, when we enter a fasting state, these fatty acids are now released and used for energy production. So we are taking our initial energy input from our dietary sources in the fed state. We are constructing a storage form of that energy, fatty acid, and then during fasting times we can release that. Cells can take that up and undergo beta oxidation reactions to regenerate ATP. Now, the problem with cholesterol synthesis is that this is not a storage form of energy. So once we form cholesterol, there's two paths for it. It is either used, and used means for membrane structure. Recall that this increases membrane fluidity. It's also necessary for many signal transduction reactions. Or it is degraded. And the degradation of this is to convert this into bile salts, and these are used for emulsification of lipids, but the energy put into the cholesterol, into the bile salts, these are not recovered for energy. There is no energy harvested. So that means the energy that's going into cholesterol synthesis, if the cholesterol isn't used for either membranes or, of course, steroid synthesis, for some biosynthetic reaction, the cholesterol is degraded and we lose that energy that's been put into the cholesterol. So that means we need a very tight regulation system in order to control cholesterol synthesis. So there's two different paths that we can obtain cholesterol. Remember the biosynthetic pathway, which is what we just looked at here. The other pathway is from LDL receptor uptake, which we saw on the last video. So synthesis is biosynthesis. LDL receptor uptake is taking what is in the environment, into the circulation, into the liver cell, and that is a form of taking up cholesterol and degrading it or repackaging it. Now, synthesis is what we need to control primarily because of the energy that we put into it. So in order to control synthesis, we have to look at what's called the SRE BP pathway. This stands for sterol, regulatory, element, binding, protein. Sounds intimidating, but it's really a rather simple system, simple and elegant system of controlling cholesterol synthesis. So what we have to start with here initially is we have to look at the endoplasmic reticulum membrane. So if this is the lumen of the endoplasmic reticulum, this is the cytosol face of the bilipid membrane for the endoplasmic reticulum, we are going to look at a protein that is embedded in this membrane that is called the SREBP. This protein traverses the membrane and then comes back, and we have two domains on this protein. One is called a regulatory domain. The other domain is called BHLH. This stands for basic, helix, loop, helix domain. Now the other part of this system that we have to consider is another protein called SCAP. And SCAP serves as a binding protein that interacts with the regulatory domain, and this protein also traverses the membrane a number of times to make a serpentine structure, and this protein serves to interact with the SREBP protein in order to hold it in place into the ER membrane. Now, under normal conditions, this domain of SCAP releases the binding from SREBP, and then this structure here, the SREBP, now translocates to another membrane structure, which is the membrane of the Golgi complex. Once it's in the Golgi complex, there is a protease located inside the Golgi that performs a hydrolytic cleavage event to cleave the two different domains apart from the Golgi. What happens next is that this portion of the beta helix loop structure now migrates away from the regulatory domain, and it becomes accessible to another protease. This other protease is located in the membrane of the Golgi, and this protease will form a hydrolytic cleavage to release this domain of the SREBP protein, which is the basic helix loop helix structure. Once this is released, this now becomes an active transcription factor. Now, what is an active transcription factor? This is a factor that now can enter the nucleus and bind to promoter elements that are upstream or 5' from genes, if this is the gene coding region, this is the promoter element, and this promoter happens to be called the SRE, which is the sterol regulatory element. When the BHLH binding factor, the transcription factor, is released because of this proteolytic event, it migrates into the nucleus and it binds to the SRE sequence, and in doing so activates the transcription mechanism to express the gene. Now the key is, what is this gene? This gene is the gene for HMG-CoA reductase. So this controls the expression of the committed step, the enzyme that mediates the committed step into sterol cholesterol synthesis. Now recall that this is a good step to control because all steps up to this point, from HMG-CoA all the way back to acetyl-CoA, are reversible. And so by inhibiting at this point, or activating at this point, we can take the HMG-CoA, bring it into the cholesterol synthesis pathway, and then this is the committed step, so that beyond this step, everything must go down to cholesterol synthesis. Now, how is cholesterol regulated here? Cholesterol regulates itself. So the translocation of the SREBP complex from the endoplasmic reticulum to the Golgi, in order to expose this cleavage site to the protease, which is localized only in the Golgi, this is not found in the endoplasmic reticulum, this movement here is inhibited by cholesterol. So this is a case where the product is a feedback inhibition to the activation of the gene that is the mediating the committed step into cholesterol synthesis. So, as a review, if we do not have sufficient levels of cholesterol, the translocation of the SREBP will occur, translocating from a quiet state in the ER to a position in the Golgi that is now expressing the protease, which cleaves the regulatory domain away from the transcription factor domain. The transcription factor domain, which cleaved, is now subject to a second protease to release this as soluble form in the cytosol. It now is able to enter the nucleus, bind to the promoter element, which is the SRE, the sterol regulatory element, and transcribe the HMG-CoA reductase gene to increase the flux of acetyl-CoA through the cholesterol pathway and increase the production of cholesterol. Now, as that production level increases, the level of cholesterol increases, which will now inhibit the translocation of the SREBP complex from the ER to the Golgi, and by inhibiting that, the proteolytic activity is unavailable, the transcription factor is not produced, the gene for HMG-CoA reductase is no longer expressed. So the control and regulation of cholesterol synthesis is basically the control of HMG-CoA reductase gene expression. Now, that's the regulation of synthesis. Now, what about the regulation that can be mediated by LDL receptor uptake activity? Recall that on the cell surface, we have our LDL receptor, we have our LDL ligand. If it's available, will bind to this receptor. Then we will have the beginning of an internalization event. The membrane will essentially be pinching off, LDLR. Here is the ligand bound to the receptor. The next step is the full internalization into that endosomal structure that we saw in the last video. And then finally, the separation of these vesicular cargo into LDL, which is in the lysosome, and the LDL receptor, which is in the recycling endosome. The recycling endosome, of course, returns to the cell surface to restore the expression of the LDL receptor on the surface, ready to receive and bind more cargo, LDL, when it becomes available. Now, the fate of this LDL in the lysosome is what's going to regulate the expression. Recall, this has cholesterol. This cholesterol will be released from the lysosomal compartment when the LDL particle is disassembled. If there is sufficient LDL, this will block the processing, translocation of the SREBP complex. In doing so, there will be no active transcription factor, and as a result of this, you already know one part of the story. There will be decreased amounts of HMG-CoA reductase enzyme, but there will also be a reduced expression of the LDL receptor. In reducing the LDL receptor concentration, that means there's less opportunity to bind on the cell surface. So we are inhibiting the synthesis of LDL receptor, conserving more energy here because the previous internalization rounds of LDL has released sufficient cholesterol to tell the cell we have sufficient cholesterol. So two patterns to shut down cholesterol processing. One is to reduce the synthesis of endogenous cholesterol to conserve energy because we have sufficient cholesterol. The other one is to reduce the uptake of extracellular cholesterol because the cell, again, is saying we have sufficient amounts of cholesterol. This is the regulation that is mediated by the SREBP pathway. And just one more side note that I didn't mention is about the nomenclature, the basic helix loop helix structure. The reason this got its name is because the protein looks like this. We have an alpha helix, we have a loop, and we have another alpha helix. Basic means that this has a basic structure, which means that it is positively charged. Now recall, when you have the DNA helix, here's our double helix, you have what's called a major and minor groove. And that major and minor groove is formed because of the intertwining and base pairing of the bases, the nucleic acid bases. This here forms essentially a trough. Adjacent to it is another trough. And these two alpha helices, separated by this loop, fit very nicely into this trough. Being a basic charge, remember the backbone of DNA is a phosphodiester linkage, and being a phosphate group, this is negatively charged. So the basic structure here is going to be attracted to the negative charge of the DNA, and these alpha helices will lay into these grooves and form very nice complementary H-bonds and ionic interactions with the bases, and that's what gives the binding specificity. This whole region is the promoter element, SRE, which is the sterol regulatory element.